Here we go. All right. So now what it is that you need to know. Now you know this is what your DNA looks like. You know about all the molecules. Today what we are going to do is we are going to take this DNA piece that I have here on my screen. Okay. And we are going to try and make the same DNA again. Okay. Now, right now, when I wanted to do that, I simply took my copying tool. I copied the entire section and I put it down. Okay. When this is going to happen with your actual DNA, things are not so easy. So the first thing that you want to do, or the first thing that you want to probably take into consideration when you're doing this, is the fact that this DNA is highly coiled, right? Which means that it is not in a shape where you can access all the parts of this DNA. So the first thing, okay, the basic thing that you want to do at this point is remove it from this helical structure and get it into something that is more linear and easier to access. So the first thing you want to do, okay, so I'm going to put one over here. The first thing I'm going to do is I'm going to unwind my DNA. When I say unwind, I want to take it from something that looks like this to something that is straight. It'll be longer when it is straight, but it will be accessible. So once I want to do that, once I have my step in the mind that, okay, I need to take this piece of DNA and I want this strand, okay, so let's say from here to here, move out and turn straight like this. Okay, so I don't want it to be wound around my DNA piece in this manner. Instead, I want it to be straight in this position. Okay, now can you see? It is so easier for me to see all the parts of this DNA, and it's just so much easier for me to just do whatever it is I would want to do. Maybe after some time, what I could also do is I could get both of these strands to be nice and straight like this. And now I can just do whatever processes I want to do. The enzyme that is going to help me in opening up this wound up DNA and making it linear is called as my helicase. Okay, so I'm just going to take this. I'm going to put this color over here. I'm going to say the enzyme that helped me open up my DNA. Okay, it literally just goes and opens your DNA like it would do for a zipper. Therefore, we also say that your helicase is an unzipping tool. Okay, it just unzips your DNA. Okay, now using this helicase, what I'm going to achieve is the opening of my DNA. Once you open up your DNA, you get two thong-like structures. Okay, so now earlier your DNA was present something like this. Now it is opened. When it is opened like this, it has a different term to it. We say that you have achieved something which is called as a DNA fork. Okay, so each one of these strands now together are called as DNA fork. Okay, once this DNA fork is prepared, you're going to use this DNA fork for the process of replication. So in certain contexts, you're also going to say this is not just a DNA fork, but it is a replication fork. This is where replication is going to take place. Obviously, you're going to have some point of origin where the replication is going to begin. So we call this as origin of replication. Okay, this is where your replication of this entire DNA is going to start. Okay, now when you say that, okay, you want to take this DNA and you want to copy it, okay, it is a very complicated process for a small molecule to carry out all by itself. So what you want to do is you want to bring in a lot of molecules in order to help you do that. So what we have done by far is we've taken this one DNA like this. Okay, so we went over, we took this DNA piece, okay, and we simply opened this DNA Okay, so I'm just going to take this in this manner. Okay, what was the molecule that helped you do that? The molecule that helped you open this particular piece of DNA was nothing but your helicase. So we are just going to put this particular label over here. Okay, just give me 1 second. You definitely want to remember the names of each and every enzyme that we are going to use here. Okay, you also want to remember the function that they carry out so that you can write it in the exam. So your helicase enzyme has now gone ahead and it has opened up this DNA for you. Okay, now this opened up DNA, you are going to take and you're going to start the process of replication for this particular piece somewhere. The position where you are going to begin this, okay, so let's say on this strand, you're going to start with the process over here. Okay, so this point where you're going to begin the process of replication is going to be called as your origin. So we're going to write origin. This is the point of origin for the process of replication to begin. Okay, now when you want the process of replication to begin, what are you exactly looking for? You have all these different kinds of nitrogen bases. Okay, so there are so many different kinds of nitrogen bases with you. All these nitrogen bases specifically have a particular kind of group that they fit in with. Okay, so if you're looking at this, I'm just going to make few in this manner. Okay, so that we can say that they're all going to fit together nice and well like this. Okay, now all of these different structures that you see onto my screen right now are nothing but your nitrogen bases, and they're just going to fit with each other. So when you want to copy this, okay, when you want to make a replica of this, you want to have a strand that is going to be present around here. Okay, so this is the new strand that you want to make, and it should have these complementary pairs made on top of it. So it needs to start with something. There needs to be some kind of enzyme that comes and starts this process. Where is it going to start this process? It is going to start that process at origin. So a enzyme, okay, or a molecule called as primase. Okay, so I'm just going to bring this here. I'm going to take its name and I'm going to write it here. This is called as primase. Okay, remember, every enzyme can be identified by the term ase that is associated with it in the end. Okay, primase is an enzyme that you are going to use to add small primers to your DNA. What is a primer? A primer is nothing but a molecule that is going to be used as the beginning for this process of replication. So you're going to have this primase molecule that is going to come in, okay, and the DNA primase is going to generate very, very small RNA primers on each of the strand. Right now, we're just looking at one strand, okay? So what you're going to get as an outcome is one primer, okay? This is your one RNA primer that has been generated. It has been added over here. This particular primer is then going to be taken further. Okay, so now you have laid the foundation for something that you want to make. This is going to go in a particular direction, and new molecules of your nitrogen bases are going to get added. At this point, you want to remember the structure of your DNA one more time. And I want you to remember that we drew this part where we said you're going to have 3' to 5' direction, right? Your DNA is always going to have its movement in 5' to 3' direction. So whenever the process of replication is also going to begin, it is also going to begin always from 5 prime to 3 prime. It was easier for this strand because this strand was already oriented in 5 prime to 3 prime direction. Things are not so good for this other strand because this other strand is anti-parallel and it has its 5 prime end at the bottom and the 3 prime end at the top. So it is running in the parallel but anti-parallel direction, okay, parallel but opposite as to what you already started. So while the first primer is going to be present over here, and it is going to begin the process of addition of a primer on one strand from this side, it is going to begin the process of addition of primer onto your other strand from a different direction. Okay, so we're going to copy this. We're going to paste it here. Okay, and once you paste it, you're going to see that it is going to be starting in a different direction like this. Okay, now when you say that it is going to start in a different direction, you again want to make sure that it is going in the direction you want. So now you have one primer that is going in 5 prime to 3 prime direction. You have another primer that is also going from 5 prime to 3 prime direction. So you seem to be doing good over here. Okay. Now you're going to run into one more trouble. The primer is going to just put one RNA primer over there. It's not going to extend those molecules. The nitrogen bases still need to be added. So you need another enzyme that is going to bring in those different kinds of nitrogen bases. That particular enzyme, which is going to do the process of bringing in the different nitrogen bases, is called as your DNA polymerase 3. Okay, this is a very important name. I need you to surely note it down somewhere. Okay, so what is this name? DNA. So I'm going to write it over here. It will be visible when I make it a little larger. Okay, this is called as your DNA polymerase 3. What is your DNA polymerase 3 going to do? DNA polymerase 3 is going to bring in more nitrogen bases, and it is going to add those nitrogen bases onto the strand one by one. But now these nitrogen bases are not the same ones that you have here. The nitrogen bases that you are adding are anti-parallel. So at the end of this entire process, okay, let's say all of your molecules of enzymes have come together. You have your helicase that has opened up your DNA. You have your primase that has come and started the process of replication. You have your DNA polymerase 3 that has made the extension of this entire RNA. If everything goes well, you are going to make this one strand over here. Okay, this is going to be your new strand. Okay, now what it's going to look like is this. So you have the original strand here and you have a new strand that you have created. When I say new strand, it is just that small piece of information of your DNA. It's like I gave you an entire textbook and I ask you to sort of copy some portion of that textbook into your notebook. Okay, you are replicating, but not the entire information, just the piece that is important for you at that time. Okay, so you're going to take that. Now here, the only trouble is when you wrote down that information, I gave you that information in English and you probably wrote it down in Marathi. Okay, this is the only difference that you're doing when you're replicating in the DNA. The information was written as AAATU. Oh, sorry. It was written as AATCCGTTA. Okay, when you went ahead and you replicated another portion, you wrote it as TTT. Okay, so when you're writing it, you will write it at TTT. But because this is an RNA, it would be U, U, U. Okay. Then this would be A, G, G, C, A, U. So the new strand that you went ahead and created is nothing but the complementary strand. This is the same strand that is already present over here. Okay. This is what you are going to do when you carry out the process of replication for the molecule of your DNA that is in the origin as 5 prime to 3 prime. Okay, so the molecule that has the orientation 5 prime to 3 prime is going to do this. Whereas when you come to the next molecule or the next piece of your DNA, here, now the things are not as easy as they were on the other side. Okay, here the primase is present in one corner and the DNA is going on opening into the other side. So you're going to have your primase, you're going to have your polymerase 3 as well. Okay, but every time that you add a few DNA molecules, okay, you add few nitrogen bases, the process is going to stop. Why is the process stopping? Because the DNA is getting unwound in the other direction. So let's say you started somewhere on, so this is one strand, this is other strand. One side you started here, the other side you also started here. This molecule is getting replicated here, but this molecule is going to get replicated here. Okay. So after a while, you notice that अच्छा ये तो यहाँ पे हो रहा है, but ये वाला molecule तो इस side पे replicate कर रहा है. So you want to bring your primer back here and replicate this portion. तब तक ये आगे चला जाता है. Then you're like, oh my God, it's again running ahead. So the primer is going to go over and it is going to start the process here. Then again, some part of it will be replicated. By the time it has completed the process of replication, this has again gone forward. So this wants to come back again and start with the process here. So in this way, this other strand that is going 3 prime to 5 prime like this and 5 prime to 3 prime here, this strand never really finishes the portion of making those DNA molecules at the same time. That is also the reason why you call this strand, okay, the one that was following the normal course, as your leading strand. Okay, so I'm going to write over here. This is your leading strand, whereas the other side, which is not following the same course, breaks, starts again, breaks, starts again, and then again breaks and starts again, is called as the lagging strand, because it does not finish all of its work on time. Okay, now because this strand is made into smaller and smaller pieces like this, so there's small piece, then again small piece, then again small piece, then again small piece, this lagging strand is seen in sort of fragments, right? It is present in very, very tiny fragments of your nitrogen bases. These fragments were first observed by a person whose name was Okazaki. Very jobless person, okay, did not have anything else to do. So he was just trying to observe what these molecules are. And for the first time, he was like, oh, okay, there's this process that is taking place. And therefore, he named what he saw after himself. And therefore, we started to call these fragments of your nitrogen bases as Okazaki fragments. The Okazaki fragments, in order to make one linear RNA, need to be joined together. Okay, so there needs to be something that is going to join these molecules together so that they are no longer separate. Because you want to join them together, you would have to bring another enzyme here, which will do the process of joining. The new enzyme that you bring into picture is something that glues these molecules together. And this kind of gluing molecule, so I'm just going to write over here, glue. Okay, this kind of molecule that allows you to glue the nitrogen bases together is called as ligase. Ligating something means to join it. Okay, so you get in this particular enzyme, which is called as ligase. And what the ligase is going to do is it is going to join. Helicase is going to open your DNA. Ligase is going to join the DNA molecules together. Okay, so at any point of time, if your DNA gets cut, you simply bring in ligase and the ligase will join it together. If you want to cut the DNA, not open it, okay, if you want to go ahead and cut the DNA, you have another enzyme that is going to do the function. Okay, so I'm just going to take this and I'm going to put the color in here. Okay, the enzyme that is going to work as a snipping tool for your DNA is called as restriction endonuclease. Okay, remember cleaving. I'm going to write over here. Cleaving means to open. In the terms of DNA, to cleave something means to open that particular DNA. Okay, when you talk about cutting open the DNA, now onward, you're not going to use the word open or cut, but you're going to say you want to cleave the DNA. And the way that you are going to cleave the DNA is by simply bringing in this particular enzyme called as restriction endonuclease. And what this restriction endonuclease is going to do is it is going to cut your DNA molecule just like this. Okay, and now because your DNA molecule is going to be cut, okay, so let's say you wanted to cut it at a very particular spot. Okay, so we're going to go back and we're going to cut it here as well. So now can you see that there is a very nice, very crisp cut into your DNA? This cut can again be ligated back like this using your enzyme ligase. Okay, so we understood there is a helicase that is going to open the helix of your DNA. There is a primase that is going to add the primers. There is DNA polymerase 3, which is going to add the nitrogen bases to make a longer chain. The new chain that is going to be made is going to be anti-parallel. Okay, it is going to be complementary. And one strand is going to go from 5' to 3' direction, whereas the other strand, because it is ulta, is going to go from the 3' to 5' direction. Yes, but while it is going from 5' to 3' direction, while it is moving like this, okay, so this is the way that it is going to go ahead. Okay, so here, why is it going back? Okay, it is going to go in this direction, again 5' to 3' direction. It realizes that it has far more molecules to add than it already has. So it's going to skip to a new position, and that is why you're going to have this strand which is going to be made in smaller and smaller fragments like this. It is going in the direction you want. Yes, it is going from 5' to 3', but it is going in lags, and therefore this particular strand is going to be called as the lagging strand, which is made up of Okazaki fragments, and the other strand is called as the leading strand, and it does not have any Okazaki fragments at all. Okay, this is what you need to understand in order to go to the next part of this chapter, which is using these things you can make DNA. But before we go to that part, I want you to see something. So we'll stop for this particular explanation here for a minute. We're going to see something for this part, and then we are going to come back and finish this thing. Okay, so just give me 1 minute. And I have yeah, sure. Okay, so just give me 1 second. I'm just going to share my screen. Yeah.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, You would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3'. And the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one.
Okay, so this is something that you were able to follow. So there's a DNA strand that you have. The DNA strand is going to be copied. Once your DNA strand is going to be copied, it is going to move in a particular direction. And there are a few enzymes that are going to help you in that particular process, right? Now we come to the next concept that they were just talking about. Okay, now here what you have. Okay, I'm just gonna make this a little blur so we don't see all of that. Okay, so here, now you have this DNA strand, okay, that you have unwound and you have made two new copies right next to it. So there's one copy of this part over here. There's another copy, okay, which is here. When you look at these replicated portions, you can see that there is one strand that was already present in the DNA. This is your parent strand. Okay, this is the parent strand. This is also the parent strand or the original strand. And then what you did is you made this new strand, which is going in a particular direction here. Okay, now this new strand that you have created, okay, can again wind up along with this. Okay, so it can go around and make a molecule like this. Okay, or it can remain same. It doesn't really matter at this point, but you have two DNA strands now. There is one which has this portion, okay, and you have the second one which has this portion. You have one parent strand and you have one new strand. Okay, when you look at this as a single piece of DNA, you will always see that one parent strand or the original strand remains as is, and only one new strand is added. Only one new side is added. When you do this, you say that your DNA replication is a semi-conservative process, which means that when you're making a new strand, you're not making everything new. You're taking something that is already present. Okay, you're taking the original copy and you're making just some parts of it back, not the entire process. So every single time that you're going to carry out the process of DNA replication, you are going to end up with one DNA that is original and one which is a copy. Okay, this also says that every single time, okay, now let's see this. There is this one cell, okay, and it had, let's say, some kind of information with it. So I'm going to take this and I'm going to take this. Okay, this was some kind of information that was present within this cell. what happened over here when it made a new cell, okay, is that it took all of that information, okay, it made a new cell, and then it copied some of the information that was already present and made two more new copies of that information like this. Okay, when it goes and copies the same cell, okay, now first time you copied this cell and you made a new one, you're going to take this, you're going to make another copy. Now what you're going to end up with is some part of this, some part of this here, some part of this guy, and some part of this guy, okay, like this, and some more new portions. And this is going to continue. Some part of the original parent is always going to continue with the new parent, okay, or the new daughter cell. And the reason that this new part is always going to continue is because it is a semi-conservative process, because every time you copy the DNA, the parent strand is the strand that gets copied. If you cut it into half, you are going to end up with some amount of DNA that was previously present inside the body of this particular organism. And because this is how it is going to present itself, we say that every single time you go ahead and you copy your DNA, you are going to end up with some DNA pieces, okay, or some portion of DNA that you already had with you. You just went ahead and you made smaller and smaller portions of that DNA. Okay, if you wanted to also put it into more proper words, okay, I'm going to write it down over here. So you are going to say that DNA replication is semi-conservative because one strand, so I'm going to write it, one strand is from,
Ma'am,
yes,
are you sharing the screen?
yes, I am. Are you able to see it? No?
No, it has stopped.
How about now?
Yeah.
Yeah. Okay. So one strand is going to be from your original parent. Okay. And you are also going to call this as an original template model. So I'm just going to write that over here. Original template model. Okay. And one strand is going to be newly synthesized. So I'm going to write, and one strand is newly synthesized. Okay, this is what semi-conservative DNA replication means. You're going to be asked this question, okay, several times in your exams. All that you need to do is mention this portion. Okay, if you just simply talk about this, you are going to get those 3 marks. Okay, that's how many marks it's going to carry. So semi conservative process of DNA replication. Now, when you look at all of these processes, okay, when you talk about your one strand of DNA going from other strand to the second strand to the fourth strand, however many number of times you're going to copy this DNA, one thing that you always need to remember is that you are copying this DNA ultimately so that you could send it to a new cell. So you want to do this process with the entire DNA. So you're going to choose one portion at a time. You're going to finish replicating that. You're going to remove those strands, keep them outside. You're going to start at a different portion and you're going to do the same everywhere. Your eukaryotic DNA, okay, is going to start this process at different locations so you can finish this quickly. There's a lot of DNA and very small time. Interphase is a very small time. So you're going to do this, okay, at multiple portions so that you can start copying your DNA at the same time over several locations. But while all of this is doing, okay, while all of this is going on, you also need somebody to check that you don't copy the same piece of DNA twice. Because if you do that, you're going to end up making this entire DNA into your chromosomes. Okay. And if you end up making more copies of the same chromosome, it is going to lead to certain kind of abnormalities. Those are the abnormalities you definitely don't want to occur in your body. Some abnormalities to name would be your Down syndrome, okay, Edwards syndrome. Definitely not good things. Okay, you don't want that to happen. Sometimes while you're copying these DNA molecules, you might also end up having some kind of mutations. Again, mutations are not good. So you don't want any piece of DNA to be copied in a wrong fashion. So where it says CAA, you want to make sure you're copying CAA and not AAC. That would be wrong. That is something that you definitely want to make sure you're keeping constant. Now, once you've done with all of this, okay, you've replicated your DNA, तुमने सब फलाना ढिमकाना जो भी करना था कर लिया. Ultimately, okay, some of this DNA, okay, so let's say this was the amount of DNA you originally had, and this is the DNA that you copied, which is going to go to the new cell. Okay, so this is something which is sorted. You already have all of this with you. Now what you need to see, okay, is that all of this DNA, once it goes into a particular cell, not just for the process of division, but for the process of the cell to function, okay, the DNA is going to be copied again. Now look at this, okay, this tends to get a little tricky here. So now let's say this portion of DNA, okay, just that strand of DNA is the DNA portion that tells your body how to make insulin. Okay, this is the portion that tells your body the information to make insulin. The entire recipe to make insulin is written over there. So what your body needs to do, okay, is first it needs to find out which cell should have this information. Each and every cell of your body has the same DNA. It is not going to be like तुम्हारे हाथ का DNA अलग है और तुम्हारे पैर का DNA अलग है और तुम्हारे eyes का DNA अलग है. No. Every single part of your body is going to contain cells, because they're your cells, they're going to contain the entire amount of DNA. Just that your eye cells have a small portion of their DNA activated, so they work like eyes. And your brain cells have a small portion of DNA activated that codes for neurons, and therefore it works like a brain cell. Your cells of the stomach have that portion of the DNA activated that works like a stomach cell. Similar to that, you're going to have these cells in the body, and they are going to be present in your pancreas. And then the pancreas is going to have the portion for insulin that is going to be active. And every single time your body wants to make insulin, what you are going to do, you are going to take the parent strand. This is the strand on which the information to make insulin is present, and you are simply going to copy this piece. This small piece, not the entire DNA, is going to be copied in the form of RNA. Okay, now this RNA that you have here, okay, which you have simply copied in order to make a new protein, okay, what you're going to do, you're going to take this some amount of DNA, this small piece of RNA that you made, okay, you're going to call this as your mRNA, the messenger RNA. This is the one that has the information to make your protein. this mRNA is going to go out of your nucleus. Okay, so this is your DNA. mRNA comes out of the nucleus. Okay, this is your nucleus. And then what it does, it goes to a very specific site in your body, which is called as ribosome. This is what your ribosomes look like. Okay, so I'm just going to take this. Your one ribosome is made up of two subunits. This is one ribosome, but it has two parts. Okay, this is your larger subunit. This is your smaller subunit. Okay, in this ribosome, what is gonna happen? You're going to have this particular mRNA. Okay, so I'm making this as mRNA. It is going to come and fit into that small ribosome. Once it comes and sits onto that ribosome, okay, what you're going to do is you're going to bring in a very peculiar kind of RNA back. Okay, so you're going to have another kind of RNA in your body. Okay, this is called as your tRNA, transfer RNA. Okay, there are many small pieces of RNA like this that are floating around in your body. Okay, each one of the transfer RNA has the ability to carry one nitrogen base with it. So on top of it, it has like this one nitrogen base. अब इसको ध्यान देके सुनना, ठीक है? You have the entire piece of mRNA. इसके ऊपर A U C U A G G U A ऐसे अलग अलग डीएनए के बेस पेयर्स लिखे हुए हैं. Okay, now each one of this base is going to be read in a very peculiar manner. तुम इन सारी चीजों को triplets में पढ़ोगे. What are triplets? Numbers of 3. Okay, so when you want to read, you remember last time I was talking about that entire wheel of your enzymes and your amino acids, where you were going to have different kinds of amino acids and they were all going to be written in a very particular manner. So if you wanted to write, let's say leucine, which is an amino acid, you will write CUU. And if you read on your DNA anywhere CUU, your body will make leucine. If your body reads somewhere, okay, the base pairs as GGU, they will go ahead and your body will make glycine. And that's how it works. Okay, you really don't need to remember the chart for this. It will be given to you in the exam. You don't really need to remember this. But each one of your transfer RNA here is going to have 3 of these nitrogen bases, and here there are going to be 3 nitrogen bases that need to be read. Okay, when you read them in the numbers of 3, they're called as codons. Okay, codons are read in triplets. So you have these triplet codons. If here you have AAU, here you're going to have UUA, complementary to what you have on the first side. Therefore, AAU will get the tRNA that has UUA come and join here on top. As soon as it joins here, this amino acid that was present over here will get released. ठीक है, ये दोनों आके एक दूसरे के साथ ऐसे जुड़ जाएंगे very nicely, and the amino acid that was present over here will get released. फिर second वाला codon को read करेंगे. So now it will go ahead and read the next codon. Whatever is the next codon, उसका tRNA आके बैठेगा. So CAGU, that particular amino acid will get released. पहले वाले amino acid के साथ वो जुड़ जाएगा. Again, the next one, GGU. So ye banega CCA. New amino acid will get attached over here. Again, you have AGQ. So तुम्हारे पास आएगा UCA, and then another amino acid will get attached. Like this, many amino acids will come and get attached, and then several amino acids together polymerize and make one protein. Okay, I need you to go back to your biomolecules chapter and remember that proteins are made up of amino acids. Amino acids can do a lot of other things, but one function that amino acids have is they make proteins. Okay, this is how they are going to make proteins. Your ribosome has a particular kind of RNA in it, and that RNA is called as ribosomal RNA. So all in all, you have three types of RNA molecules that are involved in this process. Something which is called as mRNA. Okay, then you're going to have something called as tRNA and something which is called as rRNA. M is for messenger, T is for your transfer, and R is for ribosomal. Okay, this is all you need to understand in this chapter. There is almost nothing more in this particular chapter. This is where it ends. You need to go ahead and read this portion in your chapter. Okay, and now that we have completed about 6 topics, what we are going to do is in the next class, we are going to take few question papers and we are going to sit and solve those questions. Okay, if you're fine with it, I have another student who is also doing the same questions, okay, the same topics. If you're fine with it, what I can do is I can take 2 of you together and we could solve some questions. So if you're fine with it, do let me know and I'll ask that student as well. So 2 of you can have different questions and you could ask like, what do you think should the answer be for this? And you can discuss that. So if you're fine, just let me know by tomorrow. Okay, so this is where we end for your chapter number 6. I'm going to share the recording with you. I'm also going to share the sort of pictures that we created. And I'm also giving you a video that I need you to watch so that you remember what this is all about. Okay, so this is going to be a comprehensive, you might say, like a detailed video that you should see so that you remember what all of this is for and how this has been carried out. Apart from that, really nothing else for you to remember, nothing else for you to understand. Okay, just keep on revising this portion and read your textbook. If you read your textbook and if you revise this, you're sorted. Okay, so this is it. This is where I'm going to stop for the day. I'm going to stop the recording now.